Good morning, children. In the previous session of this chapter, you have seen that how plants prepare their own food through the process of photosynthesis. You have seen the different steps, the materials required for this process, etc. So here in this session, we are going to observe or we are going to demonstrate some experiments to prove that all these materials are really needed for photosynthesis or not. Or we are going to demonstrate some experiments to prove all these materials are really needed for photosynthesis. So what are the materials needed for photosynthesis in plants? You can see here in the picture that sunlight, carbon dioxide, water are all taken in by the plants, and with the help of chlorophyll, the plants perform photosynthesis. And using the sunlight energy which is absorbed by the chlorophyll, carbon dioxide and water combines to form glucose, which is basically the process of photosynthesis, and oxygen is a byproduct or a waste product which will be released into the surroundings. So here in this session we are going to prove that sunlight, carbon dioxide, chlorophyll, etc. are really required for photosynthesis. So our first experiment is to prove that sunlight is required for photosynthesis. So what are the materials needed for this? We need a potted plant, black paper strips, clips, alcohol, iodine solution, water bath, beaker, burner, water, petri dish and dropper. So to start with the experiment, we need a potted plant. And we will keep the plant in a dark room for 2 to 3 days. Why do we keep it in dark room? Actually, once it is in complete darkness, the plant is not able to perform photosynthesis. So this plant will start using up the starch which is already present in the leaves. So with this process, by keeping it in the dark room, we can destarch the plant, or we can make sure that there is no starch present in the plant. Or whatever starch is there, it's all used up. Now we can go for fresh photosynthesis. So this is the experiment to prove that sunlight is required or not. So we have to make sure that there are some parts which do not get the sunlight. So for this, what we do is we cover one of the leaf with a black paper. We can use some adhesive tape or clips to adhere it to the leaf, so that that part of the leaf is not exposed to sunlight, whereas all other parts are exposed to sunlight. So that is our first step in this experiment after taking it out from the dark room. Now we will allow it to be in the sunlight for 3 to 4 hours for fresh photosynthesis to take place. And now we will see whether the photosynthesis has taken place or not. How do we prove that? If the starch is present, photosynthesis has taken place. If the starch is not present, photosynthesis has not done. So to prove whether starch is there or not, we will go for a starch test. But before we do that, we have to decolorize this leaf, because the leaf is green in color. Maybe our results are not very visible. So what we do is we will decolorize the leaf first. For that, we need alcohol. We will take ethyl alcohol. We will put this leaf in ethyl alcohol and we will heat it, because hot alcohol can dissolve chlorophyll from the leaf. But we cannot heat it directly because alcohol is highly inflammable. So what we are going to do is we will put the leaf in a test tube with alcohol, and we will keep this in a water bath or a beaker containing water. And we will heat the water. When water boils, alcohol becomes hot. And hot alcohol dissolves the chlorophyll from the leaf. Now we will, after some time, you can see that the leaf becomes colorless and alcohol turns green in color. Now we will take out the leaf and then we will wash it in water. And then we will go for starch test. For starch test, we need iodine solution, because iodine, when combines with starch, turns the color into blue-black. Usually iodine is having a reddish brown color. Once when it is combining with the starch, the color changes to blue-black. So when you use this leaf, you can see that the leaf shows this type of a result. The part that is there in the middle, which was covered with a black paper, does not show any color change, whereas the other parts show blue-black color. So what does this experiment prove? It shows that the middle part, which was not exposed to sunlight, could not perform photosynthesis, and the other parts which were exposed to sunlight performed photosynthesis and synthesized starch, which very well proves that sunlight is required for the plants to perform photosynthesis. So the next experiment is to prove that chlorophyll is required for photosynthesis. For that, the materials required are a potted plant with variegated leaves. What do you mean by variegated leaves? You must have seen the plants with some designs on the leaves, the leaves which are not completely green. Some parts are green and the other parts are maybe yellow or red or with some other colors. Some designed leaves like a money plant or the croton, you can take such a plant. So that kind of a plant we need, and then we need tracing sheet, alcohol, iodine solution, water bath, beaker, burner, water, petri dish and dropper. So to start with the experiment, we will keep the plant with this variegated leaves in dark room for 2-3 days as we told before. Why do we do it? This is to destarch the plant or to make sure that all the starch in the plant is used up. So here in the picture, you can see a variegated leaf. You know, some parts are green where chlorophyll is present, and in the other parts, chlorophyll is not present. So this type of a plant we are going to take. Now, after discharging in the dark room, we will keep it in the sunlight. We will keep it in sunlight for 6 hours so that fresh photosynthesis will take place. Now we will pluck a leaf and then we will trace it out on a tracing sheet. We want to have this design on the tracing sheet. Okay, so we will draw this design on a tracing sheet and then we will immediately put it in boiling water. Why are we putting the leaf in the boiling water? In this hot water, the proteins get denatured or the enzymes get killed so that no more photosynthesis takes place in the leaf. Then we will take out the leaf and we will conduct all the other tests as we did before. First, we will decolorize the leaf by using ethanol. We will heat it up and decolorize the leaf. So after some time, you can see that alcohol turns green in color, leaf turns colorless. Then we will take out the leaf and wash it in water. Then we will conduct the iodine test or the starch test by using the iodine solution. So take the leaf in the petri dish and then drop some iodine solution on that, 3-4 drops of iodine solution. Once when you drop, you can see that this type of a design you are going to get. You know, only some parts of the leaf turns into blue-black, whereas some parts remain the same. There is no starch present in the other parts. That is why it doesn't show any color difference. So you compare it with the tracing sheet that you already made. You know, the design on the tracing sheet very well coincides with this leaf. You know, design on this leaf. Only the green parts which are present in the tracing sheet has turned into blue-black, which very well proves again that without chlorophyll, the plant is not able to perform photosynthesis. Otherwise, you could have seen starch in the other areas also or color change in the other areas also. So here, only those parts where the chlorophyll were present are showing the color change. Other parts does not show any change in color, which proves that chlorophyll is required for photosynthesis. Now moving on to the next experiment, the last experiment, it is to prove that carbon dioxide is required for photosynthesis. We say that carbon dioxide is required for photosynthesis, carbon dioxide combines with water, but we need to prove that. For that, what are the materials required? We need 2 potted plants for this, not like before. For this experiment, we need 2 potted plants and we need bell jars, potassium hydroxide pellets, Vaseline, glass plates, alcohol, iodine solution, water bath, beaker, burner, water, petri dish and dropper. So to start with the experiment, we'll take both the plants. We will keep it in dark room for 2-3 days again to de-starch the plants. Okay, and then we will keep it in sunlight. But before allowing it to perform photosynthesis, here we have to prove that carbon dioxide is required for photosynthesis. So for one of the plants, we have to provide a condition where carbon dioxide is not present. So we are going to make the setup like this. Now the first plant, both the plants are covered with a bell jar. We will keep the plants on a plate and cover it with a bell jar and seal it with Vaseline at the base so that no air enters from outside. The plants can use only air which is there inside the bell jar. And before we cover it with bell jar, for the first plant, near the first plant, we will keep a petri dish with potassium hydroxide. What is special about potassium hydroxide? It can absorb carbon dioxide. So once we keep potassium hydroxide near the first plant, you can see that, you can understand that potassium hydroxide absorbs carbon dioxide from the air in the first one. But in the second one, carbon dioxide is present as there is no potassium hydroxide present there. We will allow it to perform photosynthesis. The setup, both the plants, all the conditions are the same in both the setup except carbon dioxide. Only in the first one, potassium hydroxide is kept. Now after this, we will pluck leaves from both the plants. Now for this experiment, we have 2 leaves. We will decolorize both of them by using ethanol and water bath. And after that, we will wash both the leaves and then we will conduct the iodine test. Here you can see that when you do the iodine test, the leaves show different results. What result you can expect here? From the first leaf, you can see that when you drop iodine, there is no color change shown by the first leaf, whereas from the second plant, the leaves show color change, the color turns to blue-black. So it shows that in the second one, starch is present, in the first one, starch is not present. Why starch is not present in the first one? What is the difference in the situation in both these plants? You can see that in the only the first one, potassium hydroxide is kept and potassium hydroxide absorbs carbon dioxide. So the plant, it is very clear that the plant is not able to perform photosynthesis because there is no carbon dioxide present. But the second one, all the conditions are there, so the plant is able to perform photosynthesis and the starch is present, so the color is changing to blue-black, which again proves clearly that without carbon dioxide, plants cannot perform photosynthesis. It is again an essential condition for photosynthesis. So here you can see some questions. You can go through these questions and answer these questions and bring it when you come back after the holidays. Okay. So go through the textbook and learn the lessons well. All these, try to answer all these questions on a sheet of paper and bring it when you come back after the holidays. Thank you.